Hi, this is Miss Litton, and this is Honors Bio 7th period review of Unit 5. Say hi.
Hi.
If you haven't already heard, I spoke a few words on Sunday, trying to make tomorrow a better fun day. And what is the word?
Harder test.
You still will do your group part like group. Okay, so that part's still same. And you want to think about who you want to work with on the group. I try to not spring it on you too much because I wanted to give you a few days to kind of figure out who you wanted to work with in your team and have that discussion. Do not fear. I keep seeing postings like, and the antisocial kid says what? And so you know that I would never have you work alone, right? Unless you did not study and said you hated somebody, then I'd be okay, you're on your own. Okay, but if you're studying, you're obviously going to find somebody to work with, and I'll help and facilitate that process if you need help with that, okay? Any questions on the what we're going to do or the how we're going to do it? Okay, so let's go over our big ticket items that we need to make sure we have in mind. Remember, I cannot reteach every detail, so you're going to need to take, to identify. What I would do is I would highlight things that I say that you know you need to address. If you are on some group shared notes, I would assign it to yourself, right? So you send yourself a note. You say at, and I would go at W. Litton OPUSD, and I'd say review the stages of development, and I would assign it to me so that I am informed that I need to review that particular topic. So it's Chapter 5, Cell Division, Chapter 22, Development, and Chapter 23, Patterns of Inheritance. So let's start with Chapter 5. And by the way, if you don't already know, there's a hard copy of this. I PDF'd it, and it is right now in Google Classroom, okay, if you want a copy of it. So the first thing I would know how to do is I would be able to diagram the cell cycle, name the parts or sections of the cell cycle, and what occurs at each of those stages. Now, I'm not going to go through everything, but just a basic part might look like something like this. This section right here would be called what? What do I call it in green? That would all be interphase, okay? And then this stage right here in blue would be M, okay? And I would divide this blue one up like this, and I would divide the green one up like this, right? What would I call this stage right here? G1. Oops, sorry. This is S, and this is G2. And what's the big thing that happens during G1? Yeah, so the cells get bigger because they just finished mitosis, right? What's the big thing that S stands for? Yeah, so you're taking your single-stranded DNA and you're now making 2 copies of it, which I just made a hideous mess, but you know what I mean. What happens during G2? Proteins are made. Good. M consists of, if I take this, what would I call this stage right here? Prophase, metaphase, anaphase, telophase. And then what overlaps telophase? Right here. Cytokinesis. Good. Division of the cytoplasm. Remember, mitosis is nuclear division. It's all about the nucleus. Cytokinesis is the rest of the cell. Okay? That's a hideous diagram, but I think you know what I'm doing there. Questions, concerns? Yes?
Would you just show cytokinesis on the diagram, or like?
I just show it overlapping with telophase. Yeah. Okay, what else on that one? We good? All right. Next one. Could you diagram a cell and the stages of mitosis with a cell that has 3 chromosomes, 4 chromosomes, 5 chromosomes, like what you were to do with your licorice? The licorice, I had you do mitosis and meiosis. But could you diagram each part? Could you say, okay, here is prophase. What happens during prophase? Let's say...
Organelles it becomes visible.
Not organelles. Chromosomes become what? Visible. So I would have to be able to tell, like I'm doing, so there's 6 chromosomes here. I would have to be able to tell that you had 6 chromosomes. What else is happening then? The nuclear envelope and the nucleolus disappear. What else is happening? Radiating microtubules. Whoops, sorry. What else is happening? Asters, okay? That's all happening during prophase. You would do a better job. What's the next stage? Metaphase. What happens in metaphase? They meet in the middle at metaphase. 1, 2, 3, 4, 5. I told you this one has 6 chromosomes. Okay, so I would say 2n equals 6, right, for the one that I'm drawing, right? I would maybe have some centrioles over here. This would be a good time to have colored pencils, right? Okay, what else would I see? I was trying to do a different color here, right? Remember the microtubules, which are going to pull them apart, right? Okay, what comes next? Away at anaphase. Right? So I'm pulling them apart. And then what would happen next? Okay. And then I would make sure I could see all 6 chromosomes. 1, 2, 3, 4, 5, 6. I would maybe identify that as a cleavage furrow. I would say division of the cytoplasm is what? Cytokinesis. I would name the stage and have any description. Okay, questions, concerns about that? And you need to be able to do the right, like this is one where I would say 2n equals 6, right? This is 2n equals 5, 2n equals 4, 2n equals 3. Yes?
Would the cleavage furrow happen during telophase?
Yes. Yeah. The cleavage furrow is evidence of cytokinesis occurring. Does that make sense? Because it's pinching off. Now, would you see a cleavage furrow in a plant cell? No, it would build a cell plate and then a cell wall. You were going to say something else before you really interrupted.
Part of the cytokinesis or telophase?
What is the cleavage furrow about? Remember, mitosis is nuclear division, right? So cytokinesis is, that cleavage furrow is just evidence of cytokinesis, which is happening simultaneously with telophase. It's what's going on in the cytoplasm. Okay? How do you feel about that? I'd say that's highly likely. Okay. Next, can you name and describe 3 different ways meiosis contributes to genetic variety? Yes, you can. What's the first way? Oh, no. What's the first way? Crossing over. But you would tell me what crossing over is, right? During prophase 1 of meiosis 1, non-sister chromatids exchange pieces, right? You would explain that to me. Increasing variety. What is the second way? Independent assortment. And that is because, what? Homologous pairs line up independently of other homologous pairs during metaphase 1 of meiosis. Okay, and the third way? Yeah, you don't know what sperm is going to fertilize what egg, right? That's the other way. Because meiosis creates haploid cells, you have to put 2 haploid cells together. What do you have the most of? Primarily the sperm, right? So you don't know what sperm, because you need to create a diploid cell, because you're doing meiosis, you need to put 2 haps together for a whole, right? And so you don't know what sperm is going to fertilize what egg. Keep going. Okay. Next, can you, uno momento. Can you, I want to continue. Okay. Can you describe the similarities and differences between mitosis and meiosis, meiosis 1, meiosis 2, cytokinesis in plants and animals? Let's attach that. Mitosis and meiosis. What's the big difference between mitosis and meiosis?
Mitosis is just an exact copy. Meiosis is variety.
Okay, so mitosis is a clone, exact copy, just like you said. In meiosis, you're taking a 2N cell and forming multiple N cells, haploid cells. 2 divisions instead of 1. And the difference between the 2 divisions, meiosis 1, it's all about separating the homologous pairs. And meiosis 2 is about separating the sister chromatids. Remember, the 2 big variety, crossing over and independent assortment, all occur also during what? Meiosis 1. Okay, cytokinesis in plants and animals, we already hit that. Cleavage furrow versus plate. Right, because they can't do a furrow when it's like solid wall. Good? Okay, next. Can you describe the similarities and differences between oogenesis and spermatogenesis? Spermatogenesis is forming what? Sperm. Oogenesis is forming what? Eggs. So remember, spermatogenesis is straight up like I taught it to you. No change. Meiosis, straight up. Oogenesis is the different one, right? And what makes oogenesis different from spermatogenesis?
It makes only one good one and then the three.
Okay, how can we say that?
Uneven.
Unequal.
Sorry.
You're there. Unequal what? Cytokinesis. Unequal cytokinesis. And why are we doing unequal cytokinesis? Because we have to provide all the food, nutrients, until the zygote implants. Yes?
So out of the 4 that result in meiosis, there only 1 good egg.
One good egg, and what do you call the other ones? Polar bodies. Okay, let's run through mitosis and meiosis now together. We ready? Let's do my arms, my legs, mitosis. The chromosome, first prophase. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules, asters. They meet in the middle at metaphase, away at anaphase, to nuclei at telophase, cytokinesis. Now let's do meiosis. We're getting ready for sex, okay? So the chromosomes become visible, the nuclear envelope and the nucleolus disappear, radiating microtubules, asters, synapses, crossing over. Then they meet in the middle at metaphase. It's independent assortment, right? Away at anaphase. Who's going away? Homologous pairs. To nuclei at telophase. Cytokinesis. Prophase 2. Metaphase 2. Anaphase 2. Telophase 2. Cytokinesis. We have 1, 2, 3, 4 cells, and they are haploid. Yes? Good? All right. Done? Okay. Chapter 22. Chapter 22. Can you name the structures of fertilization? Oh, yes, you can. Wait, let me see what I asked you the next thing. Okay. Structures of fertilization. So, here's an egg. Yes? Here comes Mr. Happy Sperm. What is he wearing on his head?
Acrosome.
Acrosome. What's inside the acrosome? Enzymes to digest the zona pellucida, which is surrounding the egg, right? And remember, the sperm membrane needs to come in contact with the egg membrane, and once that does, you have your fast block real quick, shuts it all down, and then later the slow block, okay? So now it says, can you name and describe all the stages of embryo development? Yes. Here comes Mr. Happy Sperm. He's got his acrosome, penetrates the egg, zona pellucida, and you have a diploid zygote. Do it. Diploid zygote, right? Cleavage, 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 and you form a solid ball of cells called a morula. So, fertilization, then you form a zygote, okay? Then you undergo cleavage to form a morula, a solid ball of cells. Okay? Then you pump in salt. Who follows? Water, and you form a blastula. So we had zygote, right? Morula, and now we are at blastula, right? A blastula is a hollow ball of cells. What do you call the cavity inside the blastula? Blastocele. Good. Okay? Then we're going to let out the gas to form a gastrula, which is a layered ball of cells. At first there are 2 layers, and then you have 3. Yeah. What is the outermost layer? Ectoderm. Middle layer? Mesoderm. Inner layer? Endoderm. Now we already know what those layers are going to go be, right? The ectoderm is going to be your skin. Technically your epidermis, not your dermis. Same. Okay? Your skin and your nervous system, right? Brain, all your nerves. Okay? The endoderm, remember the gastrula, when we form the gastrula, What happened to the blastocele? It went bye-bye. And now we have a new cavity. What's that new cavity called? Archenteron. That is going to be our what? Gut tube. That's our gut tube. So what's going to develop from the edges of the gut tube? What are the two systems? I can follow down my mouth. Yep, digestive and respiratory and associated organs and glands. Everything else has to come from the mesoderm, primarily your muscles and bones. Perfect. Okay? So, marula, do it with me. Marula, blastula, gastrula. Then the next layer is the nerula. And that's the forming of the nervous system. Nerula, nervous system. And this is when the notochord, which is part of the mesoderm, tells the tissue above it, what is that called when one tissue influences another? Induction. Fold in and form that neural tube. So the nerula is the next stage, and that is when you have your nervous system. All right, those are the stages of development. Next, can you name and describe the layers of the gastrula and what each will become? Boom, did it. Yeah? Good? All right, next, can you differentiate and give an example of induction and morphogenesis? Yes. Okay, induction, here is tissue A. Here is tissue B. Okay? When tissue A, I wanted that to be another color. When tissue A influences tissue B, when tissue A is influencing tissue B, that is called induction. Okay? What does it influence it with? Morphogens. Yeah. This process of this arrow, when A influences B, you're like, oh, that's induction going on, and it's using morphogens to do its induction. Morphogenesis is how this tissue changes. The tissue changes as a result of those morphogens. I think morphogens movement and induction influence. That's how I remember it. Morphogenesis is the actual movement of the tissue. It's changing the shape of that organism. Okay, you good? Can I keep going? Questions you want to ask me? Yes.
Okay, so like hypothetically, if we did have to do this, could you give like a real example? Or
Absolutely.
Okay.
The notochord influences the ectoderm above it using morphogens to fold in, and that's morphogenesis when it forms the spinal cord. The mesoderm influences the layer above it to fold in and form the lens of your eye. Okay? The process of one tissue influencing, the notochord influencing the ectoderm, that's induction. What is it influencing it with? Morphogens. What is the morphogenesis is the movement. Yes?
So like induction is like the signal that it kind of sends to
The signal itself is the morphogen. The process of sending the signal is induction. By influence you. Talk louder. Talk louder. Okay, I'm influencing you. That's induction, right? What am I using to influence you? My voice. You actually talking louder is morphogenesis. Thank you. Hashtag teaching. Okay. Did that make sense? Okay. Yes. You're like, why is she yelling at her? She's so mean. I had a point to make. I saw a hand. Where is it? Yes.
The nerula is formed through induction, right?
Wait, say that one more time?
The nerula?
Yes. The nerula is a result of induction. The process of folding in to form that spinal cord is morphogenesis. What did I use to influence? The morphogens. Okay, we good? We good? All right. Next. Can you describe how mitosis and meiosis contribute to the stages of fertilization and development? I can. How did we form the sperm and egg? Meiosis. What did all of this be through? What was that through? Mitosis. Yeah. And then later, then some more meiosis, right? Okay, good, good. Questions, concerns? Easy peasy lemon squeezy? Okay, next chapter, chapter 23. Here we go. Number 1, can you name Mendel's 3 laws and describe them as well as provide an example of each? Yes, we can. Okay, what's the first law? Law of dominance. And that means when you have a pair of alleles, that one allele can be dominant and expressed over the will of another allele, right? Are there exceptions to the law of dominance? Yes, and we will discuss those exceptions. I would know them. Okay, law of dominance. So you want to say, yes, there are exceptions. So maybe come back to that, okay? What's the second law? Law of segregation. When does the law of segregation occur? You know the exact stages it occurs. Anaphase 1 and anaphase 2. But really you're talking about anaphase 1 because that's when you separate the homologous pairs, right? Because when you do anaphase 1, you're separating big T from little t, if that's your homologous pair. Are there exceptions to the law of segregation? No, not for anything normal. That's called non-disjunction, trisomy 21, or we're getting it. We haven't talked about that. But no exceptions to that. Bad things happen. Okay, so law of dominance, law of segregation. What's the third law? Independent assortment. Law of independent assortment, right, is how the homologous pairs line up independently at what stage? Phase 1 of meiosis. Because that's the only time you have the homologous pairs, metaphase 1 of meiosis. This says that I can have a big T with a big R or a big T with a little r in my gametes, right? Big T with a big R in my gametes or a big T with a little r in my gametes. What says I won't have 2 T's in a gamete? The law of segregation, because the T's separate, right? Well, the alleles, right, are on that homologous pair. Right, so this could be a homologous pair. Right, and just like, oh, okay. Make this a little smaller. So this could be a homologous pair, and then this could be another homologous pair, right? So they could have lined up that way, or it could be flipped the other way. That's the law of independent assortment. But remember, at the time of meiosis, they're going to be like this, right? No, never mind. I didn't mean to say that. I'm sorry. They're going to be like this. So you're going to separate this and this from each other, and this from each other. And let's say this has 2 big T's. Let's say this is 2 little T's. Let's say this is little r, and this is big R, right? You're separating the homologous pairs from each other. And then in meiosis 2, you'll separate the 2 little r's from each other and the 2 big R's from each other in other cells, right? So this is segregation, separating the T's from each other and the R's from each other. That's why you won't have a gamete with 2 T's in it or 2 R's. You can have a zygote with 2 T's and 2 R's, but not a gamete, right? Okay, now, what's the exception to the law of independent assortment? If I had them on the same chromosome. Big T is always going to be with big S. Now I could get big T, big S, little r, or big T, big S, big R. But big T is always going to travel with S. That's a linked gene when they're on the same chromosome. What can break up linked genes? And when does that occur? No. Prophase 1 of meiosis. Prophase 1 of meiosis. Okay, ask your bio buddy. Are you a golden pony boy? Do you have a question on that? You need to ask him this later. Let's go. Okay, the law of dominance would tell you that before meiosis occurs, they are tall, smooth, and round. That's the law of dominance. The law of independent assortment says you could get big T, big S, little r, or big T, big S, big R, right? That's what the law of independent assortment says. Law of segregation says in the gametes you only have one T, either the big or the little. Only one S, either the big or the little. Only one R, the big or the little. Does that make sense? Because you'll have one copy of the R-ness, the S-ness, and the T-ness. Whew, that was scary. Okay, you good on that? All right. So now, can you differentiate between a somatic cell and a gamete? Yes, you can. A somatic cell is a what cell? Body cell. Gamete would be a sex cell, sperm or egg. Okay, phenotype and genotype. What? Tell me.
Phenotype is the physical.
The physical appearance of the genotype, right? Yeah, the genotype is what's in your DNA. The phenotype is the physical appearance. Phenotype, physical appearance of the genotype. Can you have two genotypes in the same phenotype? Can you have two genotypes in the same?
Big B, little b, big B.
Brown eyes could be big B, or
Big B,
little b. Two different genotypes, same phenotype. Okay. Homozygous, heterozygous. Use P's and height to tell me homo, hetero.
Little p, or big
T, height.
Big T,
Big T is homozygous, tall, right, dominant. Little t is homozygous, recessive. Big T, little t, you just say it's heterozygous. All right? Now, same, we got that. Now, we're going to see some exceptions from here down, right? From here down, we're going to see exceptions. So, blood types is interesting because that's an example of what? Multiple alleles. So, you could have, instead of just big I and little I, you could have big I A, big I B, and little I. And that's what leads to codominance. Because if you have big I A, big I B, big I A, big I B, then both the A allele is expressed and the B allele is expressed. That is codominance. Both traits are expressed. Okay. There's two ways to be a heterozygote in blood types, right? What are the two ways? You could have a big with a little or AB blood, right? Both are still heterozygote. Okay? Now, incomplete dominance. That is when both, it looks like blending. It's not completely one or the other. Not like I'm here and we're both here. Incomplete looks like blending to the eyes usually. Good idea for that is?
wavy hair.
Yeah, because incomplete dominance could be an H1, H2. If you were H1, your hair would be straight, like a 1. If you are H2, your hair is curly. But if you're an H1, H2, you are wavy. What kind of babies can two wavies have?
Wavy, curly.
Yeah, tell me, what is the proportion?
Wavy 50% and then 25%.
So 25% straight, 50% wavy, 25% curly. What's another example of incomplete dominance that we looked at? Flowers. Tell me. Red, white makes pink. Pink is the incomplete, right? It's not red, it's not white, it's pink. All right. Next, sex-linked. Sex-linked only gets dicey when you're talking about who? Males. Because females have 2 X chromosomes. It's going to play out dominant, recessive, either way. But males have only 1 X chromosome. So that's why it is an exception to the law of dominance, right? Codominance is an exception. Incomplete dominance is an exception. Sex-linked is an exception in males, right? Because they only have 1 X. Normally, you would say in order to express a recessive trait, you need how many copies of it? 2. But males only need 1. Okay? Polygenic inheritance is also an exception. Because before we said, okay, your T's. If you have at least 1 big T, you're going to be tall. But polygenic, poly means what?
Multiple.
Multiple genes. Your intelligence is not a factor of one pair of alleles. That would be terrifying. You have T's and R's and Q's and P's and L's and M's and N's. You could have some incomplete dominance and codominance, and they're polygenic. That is giving you intelligence. We are not tall or short, okay? We have all different levels of height. Our skin is not black or white, okay? There are all different levels in there. Those are examples of polygenic inheritance, and those are the ones that are most susceptible to environment influencing it. Okay? Can we let it go? Let it go. Let it go. Can you identify and give specific examples of multiple alleles? Yes, you can. Give me one. Oh my gosh, we just did it. Blood. Keep your brain in gear. We have a few more minutes together. Let's full force here, okay? Can you give an example of incomplete dominance? Wavy flowers. How about sex-linked trait? Colorblindness, sure. Codominance.
Wait, no. That's the blood type.
Not skin color. Blood. AB blood. Codominant. Huh? Okay. Polygenic. Skin. Height. Intelligence. Got that? That was a harder pull than I expected. All right. Next. 18. Can you do Punnett squares with one trait and give genotypic and phenotypic ratios? With 2 and 3 traits? Yes. What would you do? Multiple Punnets. And just remember to what them? Multiply. Because you're saying, can this happen at the same time? Okay? Chances. Multiply them together, right? Chances. Multiply together. All right. Can you explain a test cross and why it is necessary?
To find out if it's homozygous or heterozygous.
I am trying to find out the genotype of a dominant phenotype. I know the genotype of a recessive phenotype, right? I do not have to test a blue-eyed person. I wonder what they are, cross them with a brown-eyed person. No, they have blue eyes, so I know they're what? Little b. But a brown-eyed person, I don't know. They're not a heterozygote. I don't know. They could be a homozygote dominant or a heterozygote. That would be an unknown genotype. So who do I cross the brown-eyed person with?
Recessive blue-eyed.
Yeah, the recessive blue-eyed. And I look at their offspring, and that will give me the answer. You okay with that? All right. And then here. Can you do all the practice problems? And I have another one. It is not a slide of this. There is no slide of this. I'm going to tell you right now. Bonus help right now. Could you draw a pedigree? Am I spelling it? 3 generations in the pedigree. Could you show me a dominant one, a recessive, and an X-linked? Could you draw it? Yeah, like a dominant trait. Like if you said this, and they had 2 kids. That's 2 generations right there, right? Then I need to marry them and have, see how I have 1, 2, 3 generations? What would you need to color to reflect a dominant trait, a recessive trait, and an X-linked trait? How would it look different?
Dominant can't skip a generation.
Okay, so that's key. Dominant cannot skip a generation. Boom. What about recessive?
Recessive
Can skip generations. Good. Sex-linked. What do you know about sex-linked?
More common males.
More common in males, and if a girl has it, so too must her dad. For a girl to get a sex-linked trait, she has to get 2 bad X's, right? She has to get 2 bad X's. We don't know. Her mom could not have it because her mom has 2 X's, so she just is a carrier, right? But for a girl to have it, her father must have it. So you need to convince me with a pedigree, okay, 3 generations, that you know that you're saying this is what a dominant one looks like. This is what a recessive one. Do I want you to do a dominant one and it just looks like this?
It works.
You think you're going to get all the points? No. Okay. recessive. Nobody's sick. You do. Yeah. No. What you're trying to do is show me what I need to look for. Okay, I saw a hand. Yes.
So for just like solving X-linked, do we just assume that it's always recessive?
Yes, I've told you that. I'm not saying that to be a itch about it. What I'm saying is I will not give you any sex-linked trait that's not a recessive one. It's too hard to tell with dominant sex. There are dominant sex-linked. I just won't give you any of them. Okay, and do your additional objectives, right? And find a friend, right? Find a friend. I love you. Make good choices.